Hello everyone. In this video, we are going to be going through the first 5 questions of this life science practice test. So we'll start by reading the question and the answer, and then we'll go through and discuss why the wrong answers are not correct, and then we'll discuss why the correct answer is correct. So starting with question 1, it says a hamster kept as a class pet has a litter of 14 babies. Once the babies were 1 week old, the students monitored their growth by weighing them every other day and recording their individual masses to the nearest gram. The student also used calculators to determine the average mass of all of the babies each time they were weighed. Many of the students reported the average mass to the hundredths of grams. Which of the following best explains why the averages should have been reported only to the nearest gram? So in this case, why are we only reporting to the nearest gram as opposed to the hundredth of grams? So A says it is simpler to graph data points that are rounded to the nearest whole number. B, a calculation cannot appear to be more accurate than the original measurement. C, round to the nearest whole number makes it easier to determine which students performed the most accurate measurements. And D, the sample size was too small to justify averages in the hundredths of grams. So let's start by looking at the wrong answers first and explain why those are wrong. So A is not correct. So A says it is simpler to graph data points that are rounded to the nearest whole number. While it may seem easier to our eyes, it is just as easy to create a graph with whatever units you want. So it really doesn't make sense that that would make a difference. For the most part, words like simpler and easier in a question are not going to be correct. We are not looking for the simplest answer, but we're looking for the correct answer. So B here is the actual correct answer, so we're going to skip that and come back to that one. So C is incorrect. Rounding to the nearest whole number makes it easier to determine which student's performed the most accurate measurement. So that is not correct. The purpose of the experiment is not to compare students' results or see which student is most accurate. We want to see what the growth is, and we need all the data points in the same units. So D is not correct. So D says the sample size was too small to justify averages in the hundredths of grams. Sample size is irrelevant to how you determine your number of numbers after a decimal. As long as you're consistent with how you started, that's what's important. For example, we would never want to start measuring to the thousandths and then decide to only go to the whole number of grams. That would be just as confusing and not give us the most accurate data. The way we decide on the decimals is through something called significant figures, which leads us to the correct answer of B. So let's go back and look at B, and then we'll talk about what significant figures are. So B says, so the answer choice of B here says a calculation cannot appear to be more accurate than the original measurement. So this is the correct answer. A calculation cannot appear to be more accurate than the original measurement. This means we would have to be consistent when using numbers, as I said in the explanation of why D was incorrect. And how we do that is through significant figures. So I'm going to flip over now so we can talk about what significant figures are and give ourselves a refresher here. So these are kind of the basic rules of significant figures you can see in this picture here. So the first rule says all non-zero numbers and any zeros contained between non-zero numbers are considered significant digits. So in this case, this first example, the 3 is a non-zero number, so that's significant. The 4 is a non-zero number, and the 2 is a non-zero number. So that's 3 right off the bat, 3 significant digits. And it says here, and zeros between non-zero numbers. So all those zeros between the 3 and the 4 here, those are all going to be considered significant. So in this case, every single number is significant. Every non-zero number, and then zeros between non-zero numbers. So the second rule says leading zeros don't count. So in this example, any zero that comes before a non-zero number is not considered significant. So all of these zeros would not be significant. The only thing that's significant here are our 2 non-zero numbers, 3 and 4. So we would have 2 significant digits. So the third rule says trailing zeros count if there is a decimal point. So in this case, our number here is 0 decimal. So in that case, all of the trailing zeros are going to be significant. So the 2 and the 5, obviously, because those are non-zero, and then the 2 trailing the 5 are significant here because there is a decimal point. Now, say we had 2,500, no decimal point, just 2,500, those 2 zeros would not be significant because there's no decimal point. So trailing zeros are only important or only significant if there is a decimal point. And then as we saw in the rule before, leading zeros never count. So the last rule here says trailing zeros may or may not count, this is what we just talked about, if there is no decimal point. So just as we said before, if we have the number 2,500, that number would not be considered, those zeros would not be significant because there's no decimal. Just as the example here, we have 190,000. All those zeros at the end do not count. They're not relevant because there's no decimal point. Now, if you saw this 190,000 decimal, even if they didn't put anything after the decimal, if they put a decimal, all of a sudden, all of those zeros become significant. So you really have to pay attention with significant digits if they have a decimal point or not. If you want more refreshers on significant digits, there are 2 videos that I've linked right here. So this is a good refresher, and then here's an additional one that I posted that I think is another good review on significant digits. So I want to go back to the question one last time. Now that we've talked about what significant digits are, I'm going to go back over to our question here. So the question they're asking, the students' measurements, so they say there's a litter of 14 babies. Once the baby's 1 week old, they start measuring their growth and recording their data to the nearest gram. So they're starting out measuring to the nearest gram. The students also use calculators to determine the average mass of all the babies each time they were weighed. Many of the students reported the average mass to the hundredths of grams. Which of the following best explains why the averages should have only been reported to the nearest gram? So why are we just reporting to the nearest gram? And a reminder here that our correct answer is B. A calculation cannot appear to be more accurate than the original measurement. So if the original measurement was done in grams and all of a sudden we're reporting to the hundredths or thousandths, it's not going to line up. It's going to look weird. It's not going to be very accurate. We want to keep our numbers consistent. And in science, we use significant figures to keep, or significant digits, you'll hear both of those terms, in order to determine how many we should use. All right, so let's move on now and look at question number 2. So question 2 says, in an effort to increase the number of students eating vegetables at lunch, the school cafeteria manager asked students to sample 6 different vegetables, with each vegetable presented in both raw and cooked. The cafeteria personnel records the number of students who said that they would eat the vegetables, if presented at lunch, in each tested form. Which of the following types of graph will be most effectively show the data? So then we have A is a scatter plot, B is called a stem and leaf plot, C is a line graph, or D is a bar graph. So again, we're going to look at the wrong answers first and talk about why those are wrong, and then we'll talk about the correct answer here. So basically, what they're asking us to compare is, for each vegetable, we want to compare whether students like it raw or cooked better. So we're going to be comparing vegetables is going to be our variable here that we're going to be looking at along our our y-axis. And then the number of students is going to be on our x-axis. So let's go back and refresh what each of these looks like. So I'm going to scroll down here now to my next picture. All right, so this right here is a picture of a line or of a scatter plot. So basically what you do is you plot all of your data. You can see it's scattered, and then you're going to draw a line through all of those that show the general trend here. So scatter plots are used, as we just said, to track trends, but the key here is of numerical variables. You can see here on the bottom we've got years. These are all numbers in this example that they have on this graph. But the idea is that these are going to track numerical variables. We are trying to track vegetables. So we can't really do dots here to track vegetables. That's not a numerical variable. So now let's go and look at B, which is a stem and leaf plot. So this is a picture here of a stem and leaf plot. This is basically used to organize your data, but it isn't necessarily a graph. It's also used more for series of numbers. As you can see, they're tracking series, which is not what we're after here. We're wanting to compare, again, vegetables in different forms. That's not numerical. This is just data. And this is looking at series, which is not what we're after. So now we're going to look at C. C is a line graph, which is also not correct. We could look at a line graph here. A line graph is used basically to show how something changes over time. So the idea is that you can track on this line how it's changing. In this case of this particular graph, we have the age of Jasmine across the bottom. That's a time. And the height, how she's growing. So the idea is you're tracking something as it changes over time. We're not tracking changes over time here. We're wanting to track, do we like vegetables raw or cooked? So again, doesn't really fit this type of graph. So D is a bar graph, which is our correct answer here. So a bar graph is used to compare discrete non-numerical variables, in our case vegetables. But again, these are non-numerical across our y-axis here. We're looking at non-numerical variables as compared to all the other types of graphs that are pretty much used to compare numerical variables, as we saw. A bar graph can be used to compare these non-numerical variables to each other. So in our case, we're wanting to check, let's say they did broccoli. We could do broccoli cooked, let's say that's pink, and we could do broccoli raw, that's blue. So we can compare those two numbers to themselves. Same with them, we could go to the next one. Let's say they did cucumbers. We could again do cucumbers cooked in pink and cucumbers raw in blue. So we can compare those variables right next to each other on one simple graph. All right, let's go back now and look at question 3. So researchers spent months in the field studying the behavior of monkeys in a particular population. In order to share their results with other scientists, the researchers should first do which of the following? A, perform controlled experiments with the monkeys in the field to support their observations. B, capture some of the monkeys to determine whether their behavior in the field is reproducible in a laboratory setting. C, submit a paper describing their observations to a scientific journal for review by their peers. Or D, invite an independent, unbiased group of scientists to observe the same population of monkeys. So let's go look. Again, let's go through these wrong answers first. So A is incorrect because when observing animals, it's best to do it in their natural environment, which is in effect something we can't control. We do our best, but it's not going to be perfect. So the idea of it being controlled is not very possible when we're studying in the field. B is incorrect because animals will not act natural in a lab. It just isn't possible. It would be like wanting to see how people interact at school and then trying to move them into a laboratory and study it. As soon as you move it, it isn't going to be the same environment, and therefore we're not getting the same results and doing the same study or experiment. And then D is not correct here. D is not correct because hopefully the group doing the experiment in the first place is an unbiased group of scientists. Anybody doing science should be unbiased. The idea should be that you as a scientist are trying to figure out the correct natural answer. So we don't need another group. Multiple groups observing is not necessary for the scientific process. So those are all the wrong answers. So C here is the correct answer. C says, submit a paper describing observations to a scientific journal for review by their peers. So C is correct because that is how we share our findings in science. We write scientific articles that are peer-reviewed by other groups to ensure the accuracy. And then those peer-reviewed articles and journals are sent out to the world so that they can be read and replicated. Other scientists can go back, read through. If they want to replicate the experiment themselves, they can do that. Peer-reviewed journals have a very specific format. So the major parts, this is just kind of a general overview, but the major parts of a peer-reviewed article are going to be an abstract. So that's your first part. This is going to be a brief summary of your article. And then you'll have an introduction, which is going to introduce the research study. Then you'll have the methods next. This is going to tell the steps that you did to perform the experiment. So again, any scientist should be able to, well, any person, but in theory, any scientist is going to be able to read your methods, and they should be able to perform the exact same experiment so that they could basically see if they could get the same results. So those methods are really important. Then you're going to have the results and the data. This is where you're going to put your raw results. This is typically going to be a data table or something like that. Then you'll have a discussion. So this is where you'll discuss that data and what it could possibly mean, what implications you might draw from it, which can lead in with your conclusion, which is where the author themselves is going to draw conclusions from the data and the graphs to support those final conclusions. And then lastly, just like anything else, you always have to have a reference section. So where you put your citations to give credit to the resources they use. Usually these are going to be found in your abstract and introduction. You'll discuss maybe why the scientists decided to research this. Here's the current research that's gone on. This is what we currently know. Maybe define any vocabulary, any of those sorts of things that are done in the abstract or the introduction. They'll cite those, and then they'll have a reference section at the bottom to go through those. All right, so number 4 now here. So they give us a picture, so this is going to be really important for us to look at. So it says, and I don't know how to say this, Astabularia is a genus of single-celled green algae that have a cap at one end that is attached to a stalk. We can see the picture here of the stalk in the root-like structure called a rhizoid. 2 species are shown above. If the cap of a cell is removed, a new cap grows on the stalk and the detached cap degrades. If a stalk from species 2 is grafted onto species 1, a new cap grows at the end of the species that resembles species 1. Similarly, grafting a species 1 stalk onto a species 2 rhizoid gives rise to a cap that resembles the species 2 cap. Which of the following is the best control for the grafting procedure? So again, we'll go back to that again, but the question specifically is asking which of these below is the best control. So A says remove the cap and stalk from an individual of species 1 and observe whether the rhizoid regrows at the base of the stalk. B says remove the cap and stalk from an individual species 1 and replace the same stalk to observe cap regrowth in morphology. C, remove the cap and stalk from an individual species 1, replace both parts with the stalk from a third species of the plant, and observe cap morphology. And then D, remove the cap and stalk from an individual of species 1, replace both parts with one stalk from each of species 1 and 2, and observe the cap morphology. So the big thing with a question like this right off the bat is it is a lot of words. So I would highly encourage you to really take your time on these questions when you're sitting in your test. Maybe mark them somehow with the idea to come back and read them again if you need to at the end. But it's going to be really important that we really just take our time and read the question very carefully, make sure we know what it's asking. All right, so again, let's go through the wrong answers first. And remember, as we just said, remember what the question is asking. They gave us a lot of words here. So the question specifically is asking for which of the following is the best control for the grafting procedures. So let's define control if you've forgotten what a control is in an experiment. The control group of an experiment is the group that does not receive the new variable, which in this case is the grafting of a new species. So we're looking for one of these answers to be removed, to not be applying the new variable. So A is not correct because it does not talk about grafting at all in here. So it talks about removing the cap, observing regrowth, but there's no grafting at all. So we haven't done anything with the experiment. And then we're going to skip B and come back to B because that's the correct answer. So C is incorrect again, because the control is not receiving the experimental group. So whatever the control group is not going to be receiving that variable. So to add a third species into the mix is the opposite of that. You're adding another variable as opposed to trying not applying the new variable. And then D is incorrect because again, we're adding variables. In this case, that would be two things being grafted onto one species. So all of those examples there would not be control groups. So B is going to be correct because we're putting the same species back onto it. So we're still grafting, which is the point of the experiment, but we're not adding a new variable or a new species here. We're keeping it the same, we're controlling it. All right, so let's go down. Our last question of this video is question 5. So prior to the 1940s and 50s, it was generally accepted that hereditable material was composed of protein. It was known that chromosomes contain both DNA and proteins, the latter of which appeared to provide the complexity required by genetic material. Experiments performed in the 1940s and 50s confirmed that genetic material was actually DNA by demonstrating both its hereditability and its importance in dictating the phenotype of an organism. Which of the following is best demonstrated by the scenario described? So A, a hypothesis should not be proposed before knowing all of the facts. B, experimental data obtained before the middle of the 20th century are unreliable. C, scientific theories are subject to change in light of new data. D, a theory must be absolutely supported by the data prior to its publication. So again, let's go through these incorrect answers first. So A is incorrect because the definition of a hypothesis is an educated guess. Hypotheses are wrong all the time. We make hypotheses and we test it and we see if we're right. That is the entire point of a hypothesis. For example, we talked about in a couple of questions ago about the idea of scientific journals and how people the hypothesis would go in your introduction. There are plenty of scientific articles that are out there where scientists put forth a hypothesis, they do their experiment, and their hypothesis is completely wrong. And that is okay. That doesn't mean they have to scrap their article. That doesn't mean they have to scrap the experiment. It's okay. A hypothesis can be wrong all the time. That's the definition of a hypothesis. It's an educated guess. We test it. We see if it's correct. So B is incorrect because a lot of what we found before the 20th century is reliable and is actually still found to be accurate today. It's just that this particular experiment was not. But there's plenty of science from the 20th century that is still accurate. And we're going to skip C and come back to that. So D is also incorrect. The definition of a theory is an idea that is believed to be true and supported by facts, but cannot be 100% proven. Theories are written about all the time in peer-reviewed journals, backed up by facts, and later turned out to be incorrect by other data. For example, gravity is technically a theory. It's called the theory of gravity. We cannot 100% prove gravity because we can't see it, but it's well accepted because the facts, we've tested it over and over and over again, and the facts seem to support it. But you cannot see gravity, so you cannot 100% prove why it is that things always go down. We just do our best. We test it, we do our best, we put forth a theory, and sometimes down the road those theories turn out to be wrong, and that's okay. So now let's go back to C, which is basically what we just said. Scientific theories are subject to change in light of new data. As we just stated above, theories change all the time based on new data. We perform experiments, we find new data support these new theories, and then scientists put forth articles explaining those new theories, and that's how science goes on. So the answer here is C. Scientific theories are subject to change in light of new data. So the idea that they're wanting you to understand here is that theories are not 100% proven. There are always more experiments we could do, and who knows, maybe down the road a new experiment may prove that theory wrong. But the current theory that's out there is put out there because that is what we have with the current information and data that we have supported. This seems to be the most accurate, thought to be true theory. So this concludes the video for questions 1 through 5. You can continue down to watch the other videos if there are other questions from this paper that you have questions on.